welcome back to Curious Live. I'm Rachel Funnell, and thank you for joining me for this discussion into the fascinating science of forensic entomology. And I'm joined by Dr. Erica McAlister and Adrian Washbourne, authors of Metamorphosis, How Insects Are Changing Our World. So thank you so much for joining me, Erica and Adrian.
Thank
Hi
you.
there. Thank you.
And before we dive in, I was wondering if you could just do a quick introduction of who you are and what you do. Perhaps we could start with Erica and then go to Adrian.
Hi, my name is Erica McAlister and I'm Principal Curator of Diptera and Cyphinaptera, or fleas and flies to most people, at the Natural History Museum in London.
And I'm Adrian Washbourne, a former BBC radio producer, now as freelance audio documentarian, I guess you could call me. And I make radio documentaries for which mainly cover the areas of science and natural history and the arts as well.
Really interesting backgrounds, and I'm interested to know how did you come together to work on Metamorphosis?
The whole idea was to come up with a series that combined a different way of telling stories about insects, really, and their importance. But most importantly, the people behind these discoveries, be it the behaviour or the appearance or the discovery of the insect in the first place, or in one case, actually predicting the existence of an insect that had yet to be found. So the idea was each episode was going to take you on a journey, an unexpected journey, which would also combine modern technology, which has been born out of a lot of these discoveries as well. And we made 2 series in the end, 10 programmes, which you can still hear on BBC Sounds, which forms the sort of kernel of the book that we've now written together.
The chapter I think that really captured me was the one about the Blowfly Detectives, and so we are here today to focus specifically on forensic entomology. So for people who aren't familiar with it, could you just introduce what forensic entomology is?
Forensic entomology is the science of decomposition, especially evolved around insects. So this is how we will study a cadaver of in various situations, and we will look at the timeline of the insects arriving at the body at the certain time. So, for example, the first ones to turn up at the scene are always the flies, and you have a succession of different insects as you go through time, because different insects will be feeding on different things. So some will feed on the skin, some will feed on soft tissue, etc. So forensic entomology is the science of studying this, to understand the timelines, to look at all the various factors involved in either slowing down the timeline or speeding up the timeline, and how we can help to put a post-mortem interval at an actual date time of when the body was left exposed and the decomposition first started.
But I mean, it can tell you so much. It can give you insights into sort of the mode and the circumstances of a death, and post-mortem modifications of the body if it's been sort of fiddled with, as well as the estimation of the time of death. And people think, oh, well, you could tell the time of death quite easily from analysing the body, but you can't. I mean, blood samples, which are the most common thing, they only work for about 2 days. So what if you come across a body that's been lying there for, more than 4 or 5 days or a week? You really have to turn to something completely different. And forensic entomology is where that comes in.
So in the Natural History Museum, there used to be dead pigs in the tower, which no one really thinks about, because this is how we'd study forensics. So he had a succession, as you were, of students. All of them did survive. I will put that caveat out there now, who were studying various different processes involved with forensics. And as technology improved, so did our knowledge of forensic entomology improve. So it used to be just looking at the insects per se and identifying them and under different things, but now we can look at the DNA, we can look at gut contents, we can use high-resolution scanning imaging systems to be able to look at the hidden parts, the pupal stage of the life cycle. So there's so many different ways that we can understand what's going on at a crime scene.
Am I right in thinking you used to have a whale compost out in the ground?
Well, because again, this works on succession. So you've got to think, as anything decomposes, you're going to get these things. And what we would do would bury. We have a little. It's called a demosterium. So it's where the domestic beetles were. So we have a little tank inside to let the beetles feast away at the flesh, the carcass of whatever bony mammal, fish, etc., bird they were doing. But obviously with whales, they're quite big. That one had to get buried for a while, a long, long time ago. My favourite ones though was elephant skulls being left on the front lawn till the public complained about the smell. It's like, yeah, okay, I can understand that.
And the title of your book, Metamorphosis, I mean, that kind of comes into this thing of being able to tell the time of death, because am I right in thinking that it's the insects that undergo complete metamorphosis that can tell us the most? So why is that?
Understanding metamorphosis helps us get a better understanding of time frame. So for example, the hidden stage, so they go from an egg to larvae to pupa to adult. And the pupa is this amazing reorganization of their body tissue. So they've basically spent their larval form as a walk-in stomach. And then just before they pupate, they leave wherever they are. So they will leave the corpse and they'll go off and they will hide in this little pupa case to reorganize themselves. So what is interesting for us in understanding forensics is what's going on in that pupa time. So previously it was completely hidden. So it may do a couple of days as pupa, it may be, weeks, depending on the conditions. So Martin Hall has been looking, along with an amazing student, they've been focusing using a CT scanner to actually look at this hidden development. So in real time, seeing how it changes. So now you can give the police, say, an estimate of a couple of hours rather than days or weeks, which is phenomenal.
And so what kind of species are we typically seeing? Say there's a body been found at a suspected crime scene and it's been a few days. What kind of species might you expect to find there?
On a really good day, with really good wind going in the right direction, a fly can sniff out a dead body from up to 7 kilometres away. So flies are going to be there at the first. They're the most sensitive. Then you'll get a period of, and different types of flies. So the first ones to turn out will be the blowflies, Californidae. And then you gradually get different types of flies. You might get some house flies, you might get some flesh flies. You'll get beetles start turning up, some moths will be there, maybe a bug. And then you'll end up, the final bit of the party is some more flies who like the bones and the skin and things like that. So you have a whole succession of these different insects.
And if the insects have already left, is there any sign of them left behind?
So when the adults have emerged and flown up, they're little people like homes, they will still be around. So we're able to look at those and we can identify what species they are from their pupal cases. Plus we can tell you, it gives us some indication of the time frame. And we can also do amazing stuff with forensics to understand how old these pupal cases are. All sorts of crazy stuff you could do now.
And do you have any interesting case studies you could tell us about where insects have been pivotal in a criminal investigation?
Well, it goes back centuries. In medieval China, in 1247, there's this Chinese lawyer called Song Xiu who wrote a training manual on criminal investigations called the Washing Away of Wrongs, I think it was called. And in it, he documents several cases. The most intriguing one, and probably the first big one, is in ancient China in a rice field where a farmer had actually been murdered by a pickaxe. It's one of the tools that the rice field farmers use. And the local mayor assembles all the local farmers and tells them to bring their tools along that they use for their daily farming activities and line them up on the pavement, in each having to stand behind their own tool. And it's a hot day. They stand there for half an hour or so, and gradually on one of the tools, flies begin to assemble. And suddenly the person who claimed total innocence is found out straight away, thanks to the insects. Forensic entomology across Asia has been used for centuries, but it took ages for this relationship between flies and their use in working out the presence of dead flesh to be used in Western Europe. It wasn't until the 1800s. And there's an extraordinary case of a family who moved into a house, and the house began to smell, and they took the chimney apart, and they found there was this tragic case of an infant who was found dead, and clearly it was a murder cover-up. And the couple were immediately arrested and convicted of murder. But it was then an innovative lawyer there who was aware of the use of insects, again, analysed the insects, and there was the timeline that was shown by the insect remains and the larvae there completely exonerated them.
It was interesting because at the time they really didn't understand about succession and how long things took, but they were able to prove enough that it was old and that this corpse had been there a while. Sadly, the child had been there a while, and so the owners were like, it's okay. So we took a little bit longer to understand how long things were going. And so the 1900s, we're really beginning to start looking at succession, which is madness because it's one of the most obvious things on the planet, and we still didn't understand it. And the UK, we were lagging behind even further. So it was only in the 1930s, 1935, that we first used forensics. And this is my favourite story because I have the actual maggots on my desk, which I don't know what you have on your desk, but I have a jar of maggots that were helped in basically committing. He was called the Jigsaw Killer. I'm a little torn about. I mean, it's a terrible story, but I'm a little torn about the. I think it was a crime of passion, a true crime of passion, and his name was Dr. Ruxton, and he and his wife lived up north in Lancashire, and they were doing very well, quite successful couple, and then jealousy kicked in, and that's when it all went a bit down. And it reads more like a fit of rage. He killed her, but being a doctor, he realised that he had to get rid of the forensic evidence. So, hence his name. He cut her up and dissolved quite a lot of her body. I know it's not nice. I mean, I'll caveat it's not nice, but it gets worse because this poor housekeeper then walked in on the scene and he ends up in the same fate as her mistress. So I know it's worse still because the children were still in the house. The bodies ended up across the border in Moffat. And Moffat welcomes you for its beautiful views. And you're like, I don't know if it meant this. But luckily for the story, two ladies were out walking and they spotted the bits of remains. I think it was one arm or something they spotted, and they called the local police. And luckily the local policeman knew of a forensic entomologist, so picked up the associated maggots around the scene. And although these maggots weren't used in the actual court case, they were used as a preliminary to give them a time frame. Because what the maggots were able to do was say, hello, we're 12 days old. If you work back 12 days, find out what the person was doing. And this was the day he was driving from Lancashire to Edinburgh to see his wife's, ex-wife's, as it were, family. So he had driven past at the exact time, so they were able to say, yep, well done, maggots, for helping us incriminate him. And the good point at the end of the story is, yes, he was arrested, he was convicted, then he was hanged. But those little maggots are, I think, some of the most important maggots in the UK. Now, when you're trained to go towards a crime scene, you are just trained to just document absolutely everything. Because who knows? When we started looking at blood, we didn't know all about the DNA. We were just doing blood groups. So it's amazing how all of the forensics has drastically increased as well.
And there was another really interesting fact that I learned from your book, which was that people used to believe that insects were actually spontaneously sort of born out of corpses. So I was wondering if you could just talk a bit about that and when it was that we stopped believing this to be true.
Oh, this is where I have to blame Aristotle. So he was like, yep, everything spontaneously generates from nothing. So what was it? It was maggots from bodies. It was fleas from straw or something like that. And this is 300 BC, and it took till the 1600s for us to actually finally go, you know what, this is absolutely not true. And it's an Italian dude called Francisco Redi. He finally proved it by putting dead snakes in a box. I just love this. And so he realized that, yes, these little maggots did come, but he was like, hold on, are they coming? What's going on? And so what he did afterwards with some of the maggots, he, instead of letting them go off and do whatever and hide them, he sealed the box. And then, therefore, the maggots pupated and the adult flies appeared. And he was like, aha, this is what's going on. Reddy was doing this before a microscope. So it was the invention of the microscope that really did start helping, because we started properly looking at both the morphology of the external but also the internal structures. And we were able to look at what was going on in the larvae. And then we could start to see the different limbs develop. And luckily, Swammerdam, who is one of the early uptakers of these newfangled microscopes, he was looking at things like butterflies, and he could see within the caterpillars development of these cells that would eventually lead to the wings, legs, etc.
And we focused a lot today about how insects have changed the face of forensics, but your book is about how insects are changing the world. So I was wondering, could you tell us about a couple of other key examples of ways in which they've shaped the way that we live?
One of them is actually understanding evolution. And Charles Darwin's theory of natural selection, which became a sort of when insects became a vital tool in providing evidence for that. Charles Darwin was it was 1859. He published The Origin of Species, and it divided people, and he was still looking for further examples to sort of make his case absolutely watertight. And he was actually a botanist. Not many people know this. And 2 years after publishing The Origin of Species, he publishes this extraordinary little book called Various Contrivances, in which British and European orchids are pollinated by insects and the advantages of cross-fertilization. This book, he's incredibly playful. He actually imagines himself being an insect and sort of working its way delicately into the nectary and how the pollination works in detail. Anyway, he gets sent one day through the post by this famous plant hunter called James Bateman an extraordinary parcel. In it are 3 star orchids. In today's money, they'd be worth about £10,000. So Bateman must have really liked Darwin in order to sort of send these absolute treasures, rare treasures. They come from Latin America, and so they were known about. The nectary was about a foot long, 11 inches, and Darwin is totally mystified. As a result of studying the intimate relationship between an insect and orchid, he realises that each insect has a delicate relationship between a particular orchid. It's a high-risk strategy, but so that there must be a particular insect that fertilises this particular orchid. Alfred Russel Wallace added further weight to Darwin's idea that there must be an insect capable with a mouth part that's capable of getting inside the nectary. 4 years later, there's an a moth that's discovered that potentially has the mouth part that's big enough to do this. The actual insect wasn't discovered until 20 years after Darwin had died, but he predicted it, and it further makes the case for the whole idea that insects and the orchids co-evolve together, and through a process of natural selection, they evolve so that each insect can develop a bigger and bigger mouthpart over many, many generations, and similarly, orchids can grow similarly larger and larger nectaries, so the two coexist to co-evolve together. And it added huge weight to Darwin's theory of natural selection.
We have some of the moths here, and the mouth parts are phenomenal. And what is quite amazing about this, and what they've led to, what they stimulated, is that they're not muscles. So everything is controlled. It's hydrostatic, but just being able to slurp up that much juice and then go to another one and another one and another one has got people thinking about how we could use it. And that's what I think one of the things I really love about this book, and about a lot of the modern scientists we've looked at, is how they take something that I look at as a functional part of an insect and they go, do you know what, that really would be good for that. And so there's lots of research developing different types of needles and all sorts of things. And this moth is quite clever in what it does.
Yeah, I mean, the moth it's a straw. So it works by sucking up the nectar from the orchid. But at the same time, it has a self-cleaning device whereby there's peaks and troughs within the structure of the straw that are both water repelling and some areas which are water attracting. And it enables, during the sucking up process, for dirt to be ejected. Now, that could then translate into potentially self-cleaning needles, so that needles could be reused again and again. And this is what they're working on, apparently. Well, they are at the moment in North Carolina, to develop self-cleaning needles using very much the same principle, hydrophobic and hydrophilic synthesized areas within what is effectively straw. But it also could be used to deliver as well, which is where the hypodermic, the needle technology comes in for potentially use on humans. And the straw itself, or the needle, is so delicate, they're actually looking now to using it for taking out genes from single cells as well. So it's that degree of nanotechnology that we're potentially talking about in the future. And it all comes from the mouth parts of these insects.
It's amazing, and we didn't really cover it in the book, but there's loads that they look at the ovipositor, the egg-laying tube of parasitic wasps, because again, they can do this master manipulation. They look at the mouth parts of mosquitoes, so the creature that most people find really bad is actually helping medicine in some ways, because again, like the proboscis is able, like the Lepidoptera, it's able to manipulate around, so it can penetrate the skin at 400 times less force, and around underneath the skin, it is able to manipulate around. So imagine developing these incredibly smart needles that are not painful, they're very site-specific, and they clean and all sorts of things. Are just the advances that can go on because of this. And these have taken millions of years to evolve, millions of years of adapting to the environment that they're within and radiating across all these different types of food sources. So we've been naive not to actually take inspiration from them. And also we've been naive to ignore any of them. So it's a real message, is that this is an amazing planet. We really have a lot that we need to save, look out, and generally be inspired by.
I mean, while we're talking about how amazing insects are, I wonder if it's not too mean a question to push you. Do you have a favourite group or insect species you could tell us about?
I'm going flies. I'm going flies. I can't not go to flies. Adrian, what's yours?
It probably is the vinegar fly, actually. We used to call it at school the fruit fly. For me, it's sort of been throughout my education. It's just been there as a tool. And then discovering more and more about it and the way it's been used to justify the whole process of, genetics and how similar also it is to the human. I mean, 78% of its genome is identical to the human. And so it's been an extraordinarily useful tool. And so for me, it's been there all my life as something in the background or as a tool to do everything from A-level biology through to working with Erica.
And just finally, obviously we've discussed your magnificent book, Metamorphosis, How Insects Are Changing Our World. If anyone would like to follow along with your entomological adventures, is there anywhere else they can go to look for further information?
On X, Flygirl NHM, and all the other Threads, Blue Sky, Instagram. Yeah, they could just look me up, and I'm trying to shout out as much about the collection and the research as possible. Obviously, you can find Adrian and I have both our series on BBC Sounds. And so, yes, I'm just pay attention. We're out there, we're talking, and obviously go buy the book.
Amazing. Well, thank you so much for your time today, Erica and Adrian. It was a pleasure discussing forensic entomology with you, and I'm sure everyone at home has really enjoyed watching along.
Thank you very much.